So guys, I didn't have time to print out a new sign-in, so we're just going to recycle the one from last week. Just check, by your name on the far right. Go.
So it's that just like right here, is okay?
On the far right.
Oh, on this side.
Far right, yeah. Okay. So let's see what's going on. Are we recording? Yes, we are recording. Okay, good. Everybody at home, can you hear me?
Yeah.
Yes, we could hear you.
All right, good. Oops, wrong. There we go. Okay, I apologize for our late start. My doctor apparently does not respect the idea that classes have to go on. It's actually not his fault. I don't know why it took 45 minutes for me to get a shot, which is weird. But anyway, so today we're kind of moving towards the end of our discussion of population biology, strictly speaking. And we're very soon going to start talking about species interactions. But we do have a couple of more topics. This is the last topic that will be on the exam Friday. Metapopulations. Okay, we actually have one more topic in population biology before we actually start doing population interactions, but we stop here for Friday's exam. So we will talk today about a more complicated structure than we've been talking about recently. If you think about what we've been doing, we've been focusing on a single group of organisms. We've been talking about their vital rates, their birth rates, death rates, and survival rates of various kinds. And we've made a number of assumptions about those things. Well, we're going to start relaxing those assumptions in today's presentation and Wednesday's presentation. Okay, we're not going to relax them too much because then you'd have to be like graduate students to really be able to have the time to follow what we're doing. So we're going to keep it reasonably simple, but we're going to give you some of the more sophisticated ways that we look at real populations. and particularly in terms of conservation biology. The next two topics for today and Wednesday are very, very important in conservation biology. They offer us tools that conservation biologists use regularly in trying to manage populations. And they offer a more sophisticated understanding that allows us not to make stupid mistakes when we try to manage populations. So the first thing for today is that we're now going to consider what we call metapopulations. Metapopulations are not a single group of organisms. They are a series of connected subpopulations. And so very specifically, we're going to be relaxing the idea that immigration and emigration don't matter. In fact, today, immigration and emigration may be much more important than births and deaths. So we'll talk about this today. So we'll first give you some examples of types of metapopulations. We'll look at how metapopulations grow or decline. We'll look at the role that dispersal plays in maintaining metapopulations. And we'll finish looking at some habitat corridors that allow dispersal to happen more readily. So first, let's talk about types of metapopulations. Last time, when we talked about stochastic population growth, remember what we were looking at? We were looking at the Channel Islands in California. It was a series of patches out in the ocean. And each of those patches had a different extinction probability because of the size of the population. So all we're going to do today is add the possibility that individuals from one of those patches could move to another patch. Okay, well, that allows us to do some interesting things. For one thing, it allows us to have another way of saving a small population. If a population is so small that it encounters demographic or environmental stochasticity and is threatened as a result, if you get some migrants from somewhere else, well, that can make a huge difference in the viability of the population. It also gives some place for the offspring to go to in populations that are really good and would tend to get really packed and overcrowded and suffer from intense intra-specific competition. So if you don't like the habitat you're in, you can always pick up and search for another one. You can disperse out. So that's emigration. Coming in is immigration. So here is a second metapopulation, again in California. This is the southern spotted owl. And what you can see here is that we have a series of blobs, and we have numbers by the blobs, and we have little lines connecting some of the blobs, but not all of the blobs. All right, well, the blob is a population, and the numbers are the number of individuals in that population, and the lines represent dispersal corridors. Okay, now it's not impossible for individuals to disperse from one population to another in ways other than depicted here. For instance, down here in Orange County, you have a small subpopulation that has 24 individuals. And over here, you have a small subpopulation with 40. Normally, most of the dispersal happens this way, from the small one to the large one of 190 in the San Bernardino Mountains, and then to the San Gabriel Mountains. What? What's that?
I think that's out in the hallway.
Oh, okay. Sorry. And then down. But it's not impossible for dispersal to happen across this way. The problem is that dispersal that way is such a low rate that it's not even put on this map. These are the common corridors. And the reason that dispersal here in Orange County from one of these to the other is so low is because, well, that's Orange County, and it's all built up, and southern spotted owls don't particularly like urban areas. So they tend to try to use more undeveloped corridors for dispersal. But you see here that there are some populations that are big, some populations that are small. This represents partly the size of the area, but it also represents the suitability of these areas for the population. The San Gabriel and San Bernardino Mountains here, which have the big populations, they just have a lot of suitable habitat. And I think you can see that in some of these subpopulations, like this one here on the central coast of California, there are only 6 individuals. Well, how much breeding is likely to go on there? Well, not a whole lot. And that little subpopulation is also at a high risk of extinction simply because it's got small numbers of individuals. Okay, the big populations have a low risk of extinction, and they're much more likely to be large populations because the habitat allows for reproduction. Yes?
The areas like lower population, there's 6 owls.
Uh-huh.
Would there be mutations like deleterious mutations that would happen from like relatives? Like
Okay, so the question is, in these tiny populations like the one with 6 individuals, would there be the likelihood of accumulating those deleterious mutations? And the answer is yes, unless you have sufficient dispersal to bring in new genetic material. That's why metapopulations are so important in conservation biology. You may only have 6 individuals here, but if you have sufficient rates of dispersal, you may not accumulate deleterious mutations. So it's really important to know what the rates of immigration and emigration are. And as I said, in some cases, they may be more important than birth and death rates. They may have more to do with population viability than birth rates and death rates. So we have this little network. This is pretty typical. Now, this is a relatively large animal. Many of the animals that exist in metapopulations are much, much smaller. You may have little mice that live in one field, and then there's a farm field, and then there's a second field on the other side of the farm field. Well, that's a metapopulation. So metapopulations are actually really common. And what we've been doing with demography and building out our models of population growth, they're fine. But if you want to know what real populations do, sometimes you need this spatial view. And you need to consider immigration and emigration. So metapopulations are linked by dispersal. Dispersal is simply the movement of individuals from one area to another. It's not the same as migration. Migration has a given endpoint. Dispersal is simply casting out in the wild blue yonder looking for something better without knowing where you're going necessarily. So dispersal can be quite wasteful. Animals can disperse and just die. Animals that are migrating have a goal in mind, so to speak. And if you're more interested in that, take animal behavior next semester. Okay, but dispersal is how organisms move from one suitable habitat to another. And it also offers them the opportunity to expand their geographic range. Okay, and if you have high population densities in your population, dispersal is a way for you to try to find a habitat with less competition. All right, so now we need to talk a little bit about what kinds of metapopulations we envision. All right, so first off, all of them are divided into subpopulations, all right, and they occupy patches of habitat. You'll remember from our discussion of populations a couple of weeks ago about distributions and dispersion, that just because your geographic range occupies all of Eastern North America does not mean that you're going to be found absolutely everywhere there. You're found in patches. And so that's why this is pretty typical of organisms. So now, when dispersal is high between subpopulations, then the whole population acts as a single unit, and it's linked by immigration and emigration. Okay, but when subpopulations are linked by infrequent dispersal, well, okay, then you can have fluctuations of the population density in each of those patches independently of one another. So you can have one patch that is quite dense at a time when another patch is declining. And there simply needs to be somebody moving from the dense patch to the declining patch in order to change that dynamic. But recognize that if dispersal is common, then everybody just more or less exchanges individuals and everybody's free to find their best life. But if dispersal is infrequent, it can be stickier. You can have independent dynamics in different subpopulations. So the first type of metapopulation we'll consider is called the basic metapopulation model. And that's simply where you have a bunch of patches. And they're embedded in a matrix of some other kind of habitat that's assumed to be poorer in quality. In fact, the classic metapopulation, where the idea actually comes from, is islands in the ocean for terrestrial animals or terrestrial plants. The ocean is inherently an unsuitable habitat for terrestrial organisms. They can't live there. So they have to disperse across the water. There are others, however, and the basic model basically considers these patches as more or less equal in quality. But if we let the patches vary in quality, we can have something called a source sink metapopulation model. And a source sink model has some subpopulations that have habitats of high enough quality to permit the population to reproduce. So the population can grow in some patches, while in other patches, the environment is not sufficient to allow reproduction. So some patches are better in quality than others in the source sink model. Now, why do we call it source sink? Because the patches in which reproduction can occur can serve as sources of immigrants for patches in which reproduction is unlikely. Also, if you have a patch that does not support reproduction, then basically individuals that go there just go there to die. So it's a sink. So source sink dynamics imply that patches are of sufficiently different quality that some allow reproduction, some do not, and the ones that can reproduce serve as sources of immigrants for the ones that cannot. Okay, so this is what I was alluding to when I said sometimes immigration and emigration is more important than births and deaths in determining population dynamics. The third one is called a landscape metapopulation model. All right, so we're talking not just about the quality of the habitats, but we're talking about the nature of the matrix in which they're embedded. If there are corridors that allow easy dispersal between some patches and barriers to dispersal that prevent movement between other patches, then you can get really interesting dynamics. It is possible that two patches of habitat separated by a river that is only, say, 100 meters across may be very isolated from one another because the organisms in the two patches cannot disperse across water. In that case, dispersal between two patches that are only 100 meters apart may be virtually zero. And dispersal may have to go upriver to the headwaters and then around the origin of that river. So on either bank, patches may be linked, but the closest patches may be directly across the river, and there's no dispersal there. That's a landscape barrier. So landscape metapopulation models have the property that the rate of dispersal is not proportional to the distance between the patches. In a basic landscape model, it's always directly proportional. The more distant the two patches are, the lower the dispersal rate. That is not true in landscape metapopulations because of the operation of these barriers. So what we see here is we've got a source patch here, and these are little frogs. I don't know why there are two colors of frogs, but here they go hopping. They're hopping through a corridor, and they're hopping to a sink patch here, and here they're trying to colonize a high quality patch that no frogs are in currently. And everywhere around this source patch, there's, I thought I silenced that. Sorry about that, guys. Everywhere around the source patch, there is this matrix of unsuitable habitat. Okay, well, now these two patches here are quite close, but there's no direct dispersal between them because the low quality habitat in between them acts as a barrier. They have to go along this corridor, okay? So the dispersal rate between these two is proportional not to this distance, but proportional to this distance, which is much greater. So there's much less dispersal between them than you might imagine. Okay, what we have to do then is examine our populations in nature and determine whether the patches are of equal quality and whether there are barriers or corridors. So here's a little cartoon trying to summarize this. On the left is your basic metapopulation model. You have 5 patches here. You have 1 unoccupied patch. And over here, the rate of dispersal between the patches is proportional to the distance between the patches. Here you have high quality and low quality patches, and that's referred to by the density of the dots here. These are presumably individuals. So this is a source population, and this is a sink population. So dispersal would happen this way, but not that way very often. You could get high rates of dispersal from a source to a sink, but little or no dispersal from a sink to a source. So one way to tell whether you got a basic metapopulation model is to look at the dispersal rates. Number 1, are they proportional to the distance? And number 2, are they about equal to what you'd expect given the two population sizes? They don't need to be equal. If one population is twice as big as the other, you'd expect the dispersal from the big population to be about twice what it is from the low population back to the big one. But are the dispersal rates proportional to distance and to population size? In a source sink model, the rates would be approximately proportional to distance, but they would not be proportional to the populations. You might have a big sink population that really has no back dispersal. So are the dispersal rates about equal, or are they greatly unequal? If they're about equal, then you probably got a basic metapopulation where all the patches are more or less suitable. If they're greatly unequal, you probably have a source and a sink. And then finally, in the landscape model, here's a really good example. You may have a source here and a source here and a sink here and a sink here. And you might expect there to be high dispersal between these two, the way there is here, because they're pretty close. But in fact, there's a barrier and there's almost no dispersal here. Dispersal has to go this way. And unfortunately, since this is a sink, there's very little dispersal that way. So look to see if the dispersal rates are approximately proportional to distance. If they're not, there are landscape barriers operating. Okay, so you'll remember that last time we talked about the extinction probability of a stochastically growing population. And that was death rate divided by birth rate raised to the power of the number of individuals in the population. Well, one of the fundamental things in conservation biology is the probability of extinction of those patches. Okay, well, all right, so that's kind of a stochastic process, but we have two other things going on, right? We have immigration and we have emigration. So actually there are two ways to lose organisms from a subpopulation. They can either die or they can emigrate. And there are two ways to gain individuals in the subpopulation. They can either be born or they can immigrate from someplace else. Okay, so stop that. Please stop. I don't know why my phone is going crazy here today, but see if I can silence that a little better. Okay. So we can calculate a probability of extinction, but it's no longer deaths divided by births. It's deaths plus leaving, the rate of emigration, the rate of death plus the rate of emigration, divided by the rate of birth plus the rate of immigration. Okay, in both cases, it's the rate of loss on the top divided by the rate of gain on the bottom, raised to the power of the number of individuals in the population. So deaths and emigration are ways to lose individuals. It's death rate plus emigration rate is the total loss rate. And there are two ways to gain individuals. You can be born or you can immigrate. So birth rate plus immigration rate equals the gain rate. So it's loss divided by gain raised to the power of the population size. And it's just stochastic extinction. It's just the same thing we were talking about last time, but we're now accounting for immigration and emigration. So if the death rate is 0.1 per year and the emigration rate, the rate of leaving, is 0.1 per year, the rate of loss is 0.2. If the birth rate is 0.2 and the immigration rate is 0.1, then the gain rate is 0.3. 0.2 plus 0.1 is 0.3. And so the probability of extinction of that subpopulation would be 0.2 divided by 0.3 raised to the power of however many individuals are in the population. So just remember, you need a scientific calculator that can do exponents on Friday. Now, let's kind of get our heads out of the math for just a second and talk about what patterns look like in the real world again. We're going to talk about dispersal limitation here. Okay, so we may have habitat patches out there, and some of them may be occupied, but some of them may be unoccupied. And if they are unoccupied, it could be because extinction has occurred, but it could also be mainly because dispersal is so low from occupied patches. Well, that's called dispersal limitation. That's often caused by these barriers to dispersal, like oceans, like mountain ranges, like rivers. Okay, and even though the dispersal rate's very low, sometimes you may get lucky, and that's how we got organisms on the Hawaiian Islands. They are the most remote archipelago in the world from a mainland, and yet they had a rich flora and fauna before the European settlement, before the human settlement, actually. I think humans got there about 1000 AD. But it's important to understand that our activities cause the erection of dispersal barriers. And so humans, by our very activities, cause greater dispersal limitation for many species. Okay. So if we build roads, wildlife may be disinclined to cross those roads. That's a dispersal barrier. People can also act as agents of dispersal. They can make it more likely for organisms to move. Aboriginal people who colonized Australia brought dogs. Dogs would probably have never made it across the Sunda Strait between mainland Asia and Australia, but hey, people help them. People also do a lot of unintentional removing of dispersal barriers. For instance, ocean-going ships take on ballast water. That ballast water has organisms in it. Those organisms live in the tanks of the ships until they reach their port, at which point they typically discharge the ballast water, spewing alien species all over the waterways that they're delivering their goods to. This is probably the single most important means of dispersal of aquatic invasive species today. Okay. Now, fortunately for us, most of these invasive species don't wind up being invasive. They wind up dying out very quickly. But some of them do, and that's a problem for us. Okay, so here's an example of dispersal limitation in a little vole. And here you have the area of a patch on the y-axis, and here's how isolated it is from the nearest other patch. And you can see that small patches here tend to be colonized if they are close to other patches, but small patches that are far away from other patches tend not to be colonized. So this is like trying to do paper basketball using trash cans, right? The bigger the trash can, the more likely it is you'll make a basket. And the closer it is, the more likely you are to make a basket. So keep that in mind, because this is the fundamental idea behind island biogeography, which we'll cover in November. But this is very clearly dispersal limitation. It's really hard to find small patches that are really isolated. It's really easy to find patches that are close to other patches. And because large patches furnish a huge target, even though they're far away, it's pretty likely that eventually they'll be colonized. So here's a similar graph for a flying species. Okay, so you see the same pattern, but notice that the details are different. It's way, way easier for even the smallest patches to be found and colonized when organisms can fly than when organisms burrow in the ground. So, hey, what that tells you is that flying reduces the effect of dispersal limitation. Being a subterranean burrower means that you have a lot of dispersal limitation. Okay, so it's the same pattern, but being able to fly means that you have a much higher probability of locating close patches. Okay, so dispersal is kind of scale dependent. Depends on the ability of the organism to move. It depends on the isolation of the patches it's trying to colonize and the size of those patches. So 2 more things that we can do here is figure out what proportion of patches should be occupied versus unoccupied. And that's simply the rate of colonization or immigration, and the rate of extinction within that patch. Okay. So the probability that a patch will be unoccupied is like, once again, the rate of loss divided by the rate of gain. Okay, and in this case, the probability that a patch will be unoccupied depends on its rate of extinction in that patch. So what's the risk of extinction per unit time? And what's the rate of arrival of new individuals per unit time? It's just the loss divided by the gain. And of course, the probability that a patch will be occupied is 1 minus that, because a patch is either occupied or not. So, all you have to do is remember that loss divided by gain is the fundamental thing that helps us understand extinction probabilities in stochastic population growth and in metapopulations. Stochastic population growth, we were just talking about births and deaths, right? In metapopulations, we're talking about births and immigration versus deaths and emigration. Okay, so this is the extinction rate. This is the probability of extinction that we calculated in the previous calculation. Okay, so it's not the emigration rate, it's the extinction rate, which incorporates births, deaths, immigration, and emigration. Okay, now let's finish this up by talking about habitat corridors. So we talked about barriers and how it affects animals and plants. Let's talk about corridors. All right, corridors are strips of favorable habitat that connect patches that can be occupied. Okay, and you're probably thinking, that sounds pretty artificial. A lot of our experiments about these things are kind of artificial looking. But interestingly, they show that habitat corridors really, really make a difference. So here on the right, you see an experiment that was done at the Savannah River Reserve in South Carolina, and they allowed some ecologists to just go out in the forest out there and whack the forest and convert it into pasture. Well, not pasture, but meadow. Okay, and the meadows are of different sizes. And so they asked, if we look for animals that depend on meadows in a forest, like butterflies, how many species of butterflies would we find in patches that are isolated or similar sized patches that are connected by a corridor that facilitates movement? Okay, and I'm not thrilled with this graph down here because, look, there's no label on the x-axis. This bothers me no end that they didn't label the x-axis. That's very annoying. But what they did was mark the butterflies in one patch and just ask, what's the probability that we'll catch that butterfly in a different patch? And so in patches that were connected, that's this green bar here on the left, it was very highly likely that they'd recapture a butterfly in the other patch because it was easy for them to move around. In the unconnected patches, regardless of their shape, so here's an unconnected patch that kind of has a bar going through it, and here's an unconnected rectangular patch, and here is an example of the connected patches that have an actual corridor. It doesn't matter if they're unconnected, the probability that you'll find a marked butterfly is very low. Corridors actually work. Corridors facilitate dispersal, and we can exploit that in conservation biology. So here is a plan by the US and Mexican governments to establish a dispersal corridor across the Rio Grande in this very isolated Big Bend region of Texas. This is before all the talk about the border wall. Populations of animals, especially, less so plants, are limited by their ability to disperse across the river. And they're limited by the quality of the habitat on either side of the river. So what the two governments agreed on, again, before the border wall controversy, was that to facilitate the survival of species like ocelot, like Mexican wolf, some of the big species that require lots of open space and lots of undeveloped land, they would establish a series of national parks in this area as a migration corridor. And so Mexico has 3, and the US has 3, and this has actually increased the density of jaguar sightings, of ocelot sightings, and mountain lion sightings in these areas. And those are just, the cats. They also increase the density of other populations as well. So this is the kind of planning that you can do once you understand the importance of corridors. Corridors have other applications also, and I guess I didn't put that in. There's a plan in Florida to establish a peninsula-long corridor for the dispersal of the Florida panther. And it's virtually complete now. So that will connect Florida panther populations in the Everglades all the way up to the large populations that you find in Georgia and Alabama in the mountains there. This kind of habitat conservation planning is really important, and it's fundamentally based on an understanding of immigration and emigration in metapopulations. That Florida panther population in the Everglades is a subpopulation that has a low birth rate, relatively high death rate, and the way that it's maintained is through emigration down that habitat corridor. So this applies to big organisms as well as small organisms, and it's one of our fundamental tools in conservation biology, and it comes right out of relaxing the assumption that immigration and emigration don't matter to population dynamics. Okay, we're done a little bit early. I think I'll show you that corridor in Florida in a later lecture. So we are done for the day. This is the last stuff that will appear on the exam. And if you have questions at home, please speak up. If you have questions in class, please speak up.
Dr. Howard, I have a question.
Hang on at home. We've got one in lecture.
Okay.
Is the corridor exactly so like
Okay, so are you talking about the Florida corridor?
Yeah.
Okay, so in Florida, there's the main ecosystem is these pine savannas, and sometimes they have lots of palmettos growing under the trees, but it's basically a pine forest that is undeveloped. So you have these pines that grow at I don't know, 5-10 meter intervals, and it's very park-like if it's been taken care of. And so you have this continuous strip of habitat from Georgia all the way down to the Everglades of these pine forests. Now they're protected in various ways. Some of them are national parks, some of them are national forests, some of them are state parks and reserves, but they all form almost continuous strip down the center of the state of Florida. There obviously are some interstates that cut across. You also have Disney World, which kind of interrupts things. But for the most part, there's very few gaps in the continuous strip of forest from Georgia all the way to the Everglades now. They didn't necessarily plant. Sometimes these forests were production forests where they would harvest timber for pulpwood typically, and so they have often either bought them up or reduced the rate of timber harvest, but just anything to reduce the disturbance and make them more natural. In some cases, it may be necessary to do things that intervene, like planting or cutting things. Okay. We had one at home.
Yes, I was wondering if you can kind of describe a little bit more on the mark and recapture homework, specifically the extra credit part, because I'm a little bit confused on how you got 50%.
Okay, well, basically it has to do with, to understand the extra credit thing on the marker capture thing, you have to understand what would make an estimated population double in size overnight. Well, it's that algebra relationship, right? It's the number of animals number of animals you marked at one time divided by the total population equals the number recaptured divided the number of marked ones recaptured divided by the total recaptured. Well, if your estimate of the population has doubled overnight, it means that you've lost. Man, I got to do something about this phone. It means you've lost half of the marked individuals, because the fraction then of marked individuals in the second sample is way too low, and that means that you're getting a much smaller fraction there. And so in order to estimate the size of the population, you wind up with a larger estimate. So if you have 10 marked individuals in a sample of 100, then you estimate that 10% of the population is marked. But if you only have 5 individuals in that sample of 100, you're going to estimate that only 5% of the population is marked. Well, that means that you're going to estimate the population being twice as big. If you need more help than that, maybe we can get together during office hours or something. Okay?
Yes, thank you. What do we just email you for office hours, or do you have like
Well,
a
I'm actually physically here most of the time during those office hours. I certainly will be here today from 1 to 3, from 9:30 to 11 on Wednesday and Friday. Might be pushing it a little if you want to come on Friday, though, since we're having an exam Friday.
Mm-hmm. Yes, I was just confused on that question, but you cleared it up. Thank you so much.
Okay. Anybody else? All right, guys. Well, I'm going to stop sharing, and I'm going to assume that we're all okay for now. And I will see you Wednesday with some very exciting stuff that I'm sure will confuse the hell out of